All right, welcome back to the deep dive. Today we're going to be tackling cell disruption.
hmm
And, specifically, I think we should really focus on physical methods for cell disruption because, you're here to learn and we want to give you, a good overview of the topic. We don't want to get too bogged down, but that's kind of the whole point of the deep dive. So we've got our notes here from some industrial biotechnology
Yeah.
textbooks and things like that. So we'll be pulling from that. And I think the best way to kind of approach this is to think about what are the main physical techniques
Yeah.
and why are they so important?
Right.
What are the general principles behind them?
Yeah. It's a great way to frame it. When we think about physical cell disruption,
Yeah.
we're really talking about harnessing mechanical forces to, crack open these cells and release what's inside. And it's a really broad approach, right? I mean, we can apply these physical methods to tons of different cell types,
Yeah.
from the simplest bacteria to, much more complicated mammalian cells. And one of the big advantages here is scalability. A lot of these techniques can be scaled up for big industrial processes, which is, a key factor in biotechnology.
So we're talking about like literally just smashing stuff.
Yeah, well, smashing is maybe a little bit crude.
Okay.
But yeah, the core idea is to use mechanical energy to disrupt the cell structure.
Gotcha. So no fancy chemical reactions or anything like that?
No, not in this case. We're focusing on purely physical methods.
Okay. So let's get into some specific techniques then. The first one that pops up in our notes here is sonication. That sounds pretty intense.
Yeah, sonication, or sometimes called ultrasonication, is a fascinating technique. Basically, you're using high-frequency sound waves, way beyond what our ears can pick up to disrupt the cells. These sound waves create these tiny microscopic bubbles in the liquid, and those bubbles collapse really violently, and it's that force, that implosion, that's strong enough to rupture the cell membranes. And sonication is great because it can be applied to a lot of different cell types, but, and this is an important caveat, scaling it up for larger volumes, it can get really energy intensive
Right.
and generate a lot of heat, which can sometimes be a problem if you're trying to extract, delicate proteins or other sensitive molecules. You want to cook them in the process.
Yeah, no, that makes sense. All right, so next up we have bead milling. This one to me sounds a bit more like just a good old-fashioned grinding process.
Yeah, that's a good analogy.
Yeah.
You're essentially putting your cells in a container with all these tiny, really hard beads,
Huh.
often made of glass or ceramic, and then you shake or stir it vigorously.
Okay.
The beads collide with the cells, smashing them open. It's a very effective method, particularly for cells with tougher cell walls. But again, there are trade-offs.
Okay.
It can generate heat, and it might not be the best choice if you're trying to isolate very specific fragile components.
Interesting. So it's kind of a balancing act,
Exactly.
choosing the right tool for the job. So then we've got high pressure homogenization.
Yes,
That just sounds like a lot of force.
it is. It's a powerful technique, especially for larger volumes. Imagine you've got this liquid with your cells in it,
Right.
and you force it through a tiny opening under incredibly high pressure. The pressure drops suddenly as the liquid goes through the opening, and that creates these intense shear forces that rip the cells apart. It's highly effective, but like any method, it has its limitations.
Okay.
You need specialized equipment to generate those high pressures. And again, it can be quite disruptive, not just to the cell membrane, but also to the stuff inside.
Right.
So you have to consider that depending on what you're trying to extract.
Okay. So then I see here in the notes we have French press. Is that kind of similar to high pressure homogenization?
Yeah, it's definitely in the same family. The French press also uses high pressure to force a cell suspension through a small opening, leading to cell lysis. But it's typically done on a smaller scale.
Yep.
It's often used in research labs rather than large-scale industrial settings. And it offers a bit more control over the process compared to the, massive forces involved in high-pressure homogenization.
Gotcha. So it's like a more delicate version?
Yeah, you could think of it that way.
Okay. And then lastly, we have liquid homogenization, or sometimes it's called high shear mixing. What's the idea behind this one?
So liquid homogenization is all about generating really high shear forces within the liquid itself.
Mm-hmm.
You're basically stirring or mixing the cell suspension super fast,
Mm-hmm.
creating these localized differences in flow velocity that tear the cells apart. It's a gentler method, often used for more fragile cells, or when you're really concerned about preserving the integrity of certain intracellular components.
I see. So we've got all these different methods from sound waves to grinding to using pressure and shear forces. It's really quite a diverse toolbox.
It is, and that's part of what makes cell disruption such a fascinating field. There's no one-size-fits-all solution. The best method really depends on the specific type of cell you're working with, what you're trying to extract, and the scale of the operation.
So, for our listener who's trying to wrap their head around all of this, is it fair to say that understanding these different approaches gives them a pretty good foundation for how scientists and engineers are actually, getting inside these cells?
Absolutely. Cell disruption is a fundamental step in so many biotech applications,
Yeah.
from drug development to biofuel production. Knowing how these different physical methods work gives you a real insight into how we're harnessing the power of these tiny biological factories.
So let's just do a quick recap. We covered sonication using sound waves, bead milling for that mechanical grinding,
Yeah.
high pressure homogenization, and the French press using intense pressure, and liquid homogenization with those shear forces.
Exactly. All critical tools in the biotech world.
Okay, great. So here's something to think about. If you imagine a delicate plant cell versus, say, a tough fungal cell, how do you think those structural differences would impact which cell disruption method you'd choose? I think it really highlights how important it is to understand the cell you're working with before you try to break it open. Thanks for joining us for another deep dive,
It's been a pleasure.
and I'll see you next time.
See you next time.
Bye.